Welcome everyone to the last day of the skill enhancement workshop on multi-omics data integration and data analytics from 27th September to 30th September 2022. This workshop is being organized by Center for Computational Biology and Bioinformatics, Amity University, Uttar Pradesh, Noida campus, in collaboration with the Ensemble Team, European Bioinformatics Institute or the EMBL-EBI, Cambridge, UK, Tauber Bioinformatics Research Center, University of Haifa, Israel, Pine Biotech USA, and Illicit Data Corp, New Delhi. Our first session for today starts with AI/ML applications in healthcare, vaccine development, and medical imaging by Dr. Kamal Rawal, sir, Professor and Head, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida. Dr. Kamal sir is a scientist and clinician working at the interface of medicine, healthcare, and artificial intelligence. He is also an academic, speaker, author, and thought leader on new technologies in medicine and healthcare. He has published in high impact journals and presented in leading international conferences. He has a deep background in practice of medicine, clinical trials, artificial intelligence, human computer interaction, technology analytics, product and user experience, and consumer insights. Welcome, sir. I now request Dr. Kamal Rawal, sir, to please start the session.
Thank you. Am I audible?
Yes, sir.
Yes, sir. The screen is
Okay.
also visible, sir.
So my talk is basically divided into two sections. First, I'll be talking about vaccine development. Uh, so our lab is on forefront of development of path-breaking technology, vaccines for drug repurposing, for drug discovery modes, network biology, and also in diagnostics. Especially, we are analyzing the lung cancer patient CT scans and their genomic data, and we're trying to correlate the relation between the cancer and the genomics data sets. To begin with, on vaccine development story, vaccine development is pretty expensive cost. You require around on an average 500 million US dollar, and it requires around 10 years of time frame where a vaccine can get into the market. Sometimes if you see failures and counter failures during the process, it can go to up to 1 billion US dollar as well. But COVID-19 has changed the rules of games. There has been sense of urgency shown by governments and regulatory agencies, which has saved time. There have been parallel execution. A lot of things are happening parallelly. For example, clinical trials are happening parallelly. The in vitro study, in vivo study, a lot of things were overlapped. One after another, things were happening. Apart from that, the regulatory approvals were coming pretty fast. So all these things have made sure that COVID-19 vaccine came into the market within 1.5 to 2 years or so. So we have developed a new platform for finding candidates, including COVID-19 with our platform. And we have tested our platform on a number of candidates, and Corbevox is one of those, which is a protein subunit vaccine manufactured by Biological E. It was developed by our collaborators at Baylor. And when we tested this candidate on our platform, the platform gave a pretty good results, characterized, found out a lot of information from there. So the platform which we have made is known as Vax AI. Briefly, it saves 3 to 5 years of preclinical studies. Over 30 to 40% of cost is saved, and 90 to 98% accuracy has been observed. Right hand side, you see a kind of a system where you have multiple layer neurons, 1900 plus neurons, and there have been bias and weight constraints, leaky ReLU functions, and batch normalization. So a lot of advanced technologies have been used to build up this system. And also, this is the architecture of the whole system, where it's a client user-based system, client server-based system. We have used Ninjix, Unicorn, Class, Celery, and so on and so forth. We also received a lot of media traction. The paper was published in 2021 September, and we started getting the news reports and news letters from Indian Express, Pioneer, and other places also. So let me just take you toward this journey of Vaxidia. So it's a web-based tool for classification of protein sequence into vaccine candidates or a non-vaccine candidate using a deep learning system. can see the link over here. It's free for academic users for a limited number of sequences. For commercial users, we have a different arrangements. For pharma companies, we have a different arrangements. Now you can call this a predictive services, which uses deep learning technologies to predict whether the given protein are potential vaccine targets or not. It uses various parameters such as biological and physical chemical properties. And it also has the web interface where you can put in the sequence in faster format and the email IDs can be entered into it. This is the overview of the workflow. We start with data collection, protein annotation. We do feature selection, we do data set preparation, pre-processing, deep learning models, hyperparametric tuning, stratified k-fold training methods and validation, testing the system and probability calibrations. So the first task is we have collected a large number of data set from positive and negative data set. The positive data set includes the antigens of the protein sequence, which has shown some sort of a evidence that it is immunogenic, it is able to produce some immune response when it is entered or injected into in vivo system like mice, human, or other organisms. So we collected data from variety of sources such as Isham in 2018, Sarom et al. In 2018, Vilela in 2019, Flower et al. In 2007. Similarly, we also collected data set from negative situations. And, in all, we have collected 384 positive data set and almost similar numbers from the control data sets also. We also divided our data set into various species by type. For example, we divided data set into protozoa, viruses, and fungi and other things. So you can see there are various positive and negative examples belonging to different classes also. Okay. Just give me a second, please. Sapti, ये आंसर करना चाहिए मेरी को बोलना सर टॉप पे बोलो सर वो मीटिंग So what we did, we had around 1,000 plus data as positive and 947 negative data set from UniProt. And we annotated those data sets into biological features using physicochemical features. We totally had 9,000 plus features. And biological feature had various bioinformatics tools were used to compute these biological features, and PROTA is a special package which was used to compute physical chemical features. And these are the examples of variety of features like subcellular localization, addition property, D values, how many cleavage site the protein has, how many transmembrane helices it has, instability index, amino acids, MHC class 1 ligands, how many high binders are there, the number of peptides generate, the molecular weight, Willis factor predictions, whether the gene was essential, non-essential, pathogenicity prediction, homology prediction, and a lot of other parameters were taken. For physicochemical properties, we had amino acid composition, autocorrelation, and so we have conjoint triad, quasi-sequence order, where we computed sequence order coupling numbers, quasi-sequence order descriptors. So these are different kind of descriptors which we use to compute the properties of a protein sequence. Now the question arose that whether all the properties are important or there are some properties which are more important and some are less important. So in order to solve that question, we used a system called feature selection, where we used a method called Welch t-test, and we computed the difference of property values, the parameter values which I just discussed, and selected those features having p-value less than 0.5. Then we prepared the data set to training and testing. And there are a number of positive samples like 355. There are negative examples also, 169. And Similarly, we have testing example. We took less number of testing because we wanted to give good focus on the training examples, but testing examples were also taken. So in this strategy, we used more like 80, 90% examples were used in the training sets, and less number in tests. But we have also used other strategies where 50% examples were used for training and 50% for testing. Similarly, there are a lot of other strategies for training and testing were also used. Next, we computed certain parameters in order to pre-process our data using the scikit-learn library. This library standardizes a feature by removing the mean and scaling to unit variance. Then we went ahead with model creation and hyperparametric tuning. This is the overview of our vexillary model. We have 1400 plus nodes. We have 2 hidden layers, 1 input layer, and there are 2 output which can produce, which states whether a given protein, when computed during the process, will be a vaccine candidate or not a vaccine candidate. Over 1900 different neurons have been used. There has been Dickey-Relu function which we use. We did batch normalization, and we also use softmax activation and constant bias initializer. Once you are doing machine learning, we need to make sure there should not be overfitting or underfitting of the model for a given data set. Because if you overfit, it's like a cramming of the story. The model will start giving you very good results on training data. But when you use this model on some independent data set, it's bound to fail because model has actually learned the parameters, kind of cram the parameters or memorize the parameters which were used during the training scenario. So in technical terms, which we call, the model will fail to generalize on unknown data, that is a new independent data. We used multiple strategies to avoid overfitting. At model level, we use regularization, constraint of weight and bias in fully connected layers of hidden layers. Then we also use batch normalization layer after activation layer in hidden layers. When compilation of model, Adam optimizer equipped with exponential learning rate decay over the period of training cycle. We also use categorical cross entropy loss with label smoothing factor of 0.2. During training, we use early stopping method, and we store the best weight when stopped early. Then when we were training, we found that the data was not balanced. So we needed to balance the imbalanced data sets. And so here we used a different strategy. Similarly, we have hyperparametric tuning was conducted using Keras tuner. And using hyperband tuner with maximum epochs is 800 and seed value of 1 for reproducibility. We use training data set to train and test the data set to validate in hyperparametric tuning. These are some of the parameters, like number of hidden layers were 2 to 10, and number of neurons fully connected layers inside the inner layer 1600 to 2500. The type of weights initializer used in fully connected layers inside the hidden layers were orthogonal, normal. We use weight normalizations, bias normalizations, weight max norm constraints, bias max norm constraint value, learning rate, and so on and so forth. Then we use exponential decay rates, Adam epsilon, the exponential staircase, and these kind of parameters are used in the. Once we did the training, we were trying to improve our model using stratified k-fold training. This technique was used because we didn't have thousands of lakhs of examples for deep learning. We had to train our model by taking one section of the data set and training our system and then putting it back to the program and also. So these are different kind of systems which were used and which we called stratified k-fold model. The K means number of times you do the trainings. So we call these as weak classifiers. Thereafter, we ensemble all weak classifiers and use them as a consensus when predicting or testing so that we can construct a strong classifier. So ensembling basically combining together different kind of approaches. So here we use consensus of various kind of outputs generated by 5 models. We also averaged out the output generated by the 5 models also. So there are, you can see this architecture of how to combine various weak classifiers. The idea of combining weak classifier to the strong one is once we have the weak model, you are getting some answers right, but you're getting most of the wrong as well. So then we make another model, which is again giving some sort of a right answers from your testing data or validation data set. So how to solve this issue is to use strength of each weak classifier and combine them to make a consensus, so that each specialist classifier 1, 2, 3, 4 is actually looking at those concepts. Make a strong classifier. Then we did the testing. We tested with 1400 plus features. We tested with 9000 plus features. We tested with 11 features, 20 features. So we had different models, and we tested with different features on which the models were constructed. So while we were testing this, we were getting pretty good results. The most accurate ones were belonging to 1400 feature model, which had both biological and the physical chemical feature. You can see it's 92 to 97% of accuracy or so. But if we take all the features without any filtering, the accuracy goes down. That means it shows that even having a large number of variables to solve your issues, it does not lead to a better solution or better answer. So always a better strategy or good strategy to filter out things which are not needed during the process. Similarly, we used with 11 feature systems, the accuracy was low. So that shows they were insufficient. We could add more numbers, and there were 20 features as well, which were giving a pretty good results, but not as good as the 1400 feature model also. Similarly, we tested different number of features and so on and so forth. And then we did the comparison between strong and weak classifiers, how they are contributing, what sort of effect do we have if we have single classifier. If we combine different classifier, what sort of effect do we have? Then we plotted the ROC curves. And if you see, the ROC curves are pretty good, ranging between 92 to 98%, and so on and so forth. So similarly, we did with different kind of consensus models and average models. We did the probability optimizations as well, and it was given by softmax function. We use these probabilities as confidence score to approximate the uncertainty of our model predictions. This is one of the common practice and reflect how we reason. The problem is that our algorithms are not intelligent enough to provide probabilities in the form of trustable confidence score. So they tend to not to calibrate result. They overestimate or underestimate the probabilities if we compare them with the expected accuracy. This results in misleading reliability or corrupting our decision policy. So that is how we use this probability distribution. These are variety of reliability diagrams, and before and after calibrations, you can see the reliability diagrams as well. These are expected calibration errors. You can see how we are computing this score. This is the formula, and this is the brief code of computing this ECE. And then we try to reduce this ECE parameter using temperature scaling, and by including the entropy and other sort of systems in the program. We did these kind of temperature scaling. And before calibration and after calibration, we started seeing some sort of a difference in some stories. There was no improvement also. Then we also plotted the reliability curves. Then we benchmarked our program against the well-known systems which are available online, like VAXI-JEN and VAXI-GNML, using the bacterial data set, protozoan data sets, viral and fungi data sets. So one of the benchmarking data set had 13 non-vaccine candidates and 25 vaccine candidates. So here we have sensitivity levels. where you have 91%, 84%, and 90%. If you see, our program is actually beating the others. And VaccGNML is pretty close. Again, it's a machine learning system, but ours is a little better, slightly better, but it's doing much better than the VaccGene system. If you take virus examples, our system is pretty much 100% accurate. If you take protozoan system, we are not performing as good as the VaccGen, and VaccGNML is much worse. You can see 50% of prediction, and also you can see the wide variety of examples, performances, and if you see some of the independent vaccines which we took during the process, and if you took this MenB, that is meningitis B vaccine, which had the protective antigens. And, there are like chain A, factor H binding protein, serial heparin binding antigens. And so the first one is 98.4 probability, factor H is 93 probability, Nesarian heparin is 97% probability. Similarly, we tested a system on DPT vaccine, which had number of toxin subunits and all. And you can see most of the time our program is able to predict them correctly, where the expected output match with the results of XCDL system. In one of the example, we could not do it, and we are trying to understand this protein and see where are the failures, what are the loopholes, and how should we improve our programs or parameters or the tuning of parameters so that we are able to improve this kind of a prediction as well. Nevertheless, we were like 85-90% correct in this scenario also. Similarly, if we talk about Mycobacterium tuberculosis, same kind of thing we observed. We were actually getting all the right answers, like 100% or so. Similarly, vaccine against Corynebacterium diphtheria, we got the right answers. So if you look at the performances on independent vaccine candidates and all, VaccGen is performing pretty bad. VaccGen is reasonably okay. VaccDL is outstandingly, outperforming all these two competitors. And this is the example of result page. It's freely available. You put your job in the server. The server gives you intermediate page and give you some answers. You are assigned job ID. Your number of sequences are also provided, and it refreshes itself and tell you whether your sequence which you have entered is a vaccine candidate or not a vaccine candidate. Deployed using Linux, Panther, Python 3, TensorFlow, Flask, Celery, Nginx, and Unicorn. And this is the architecture of deployment. And the usage is pretty easy. You enter your data into FASTA format and email ID. You get the results on web itself. You can also enter your email ID and the result and lands up into your email IDs. This is how the results look like. And each protein which you have entered will be assigned some ID and will be predicted, given a prediction. And the confidence score is also given. You can also download detailed version of these results. This is what about the AI part. The second part which we are working on, medical imaging part. This is another big project which our lab is working on. The idea is to look at the medical images like CT scans or X-rays or MRIs and try to do some diagnosis. And one of the projects which we are working with industry as well as hospitals is to look at the CT scan of the lung cancer patients, identify the properties or features of the tumors of lung cancer, and train our algorithm based upon those properties. The algorithm is designed to match the intelligence of radiologists, experts radiologists. We have tested it on 2000 plus patient data sets. And we have found the same kind of properties, same kind of explainability of our model, what a radiologist thinks of. If he sees the size of the tumor, size of the mass, the boundary of the mass, the texture, a lot of other features which he thinks what is listed in medical textbooks also. Our program also does the same kind of activity. And it does in automated way, so the output is generated in less time. Right now, we are working with radiologists. We are working to include more data sets to make it more robust and explainable and interpretable AI system which we are making. The second thing which we are doing is with our system is actually correlating the radiological images with the genomic data. The genomic data is the same patient's data, lung cancer patients. Their CT has been taken, and that CT scan is used, and the genomic data is used, and we are looking at some specific oncogenes and see what mutations can be correlated with the features which are present on the CT scan of the lung cancer patients' tumors. And we have found pretty interesting results. One of the paper is there. We published in one of the very big journal like Journal of Clinical Oncology, and then we are right now writing in Lancet. Again, it's pretty high impact, very impactful journal. And one of the demonstration I can give right now, if you see, this is a lung cancer CT scan. These are the lungs, these are the kind of backbone, and this is the chest region, these are the arms, shoulders, and all. So this red bounding box is designed to find out the tumor inside the lungs of the patient. Also, so this red bounding box is actually an algorithm which is looking at the tumor. If you see these masses, this is a mass, the lung cancer patient's tumor. You see the texture and everything is able to detect it with a 99% accuracy results also. Similarly, this mass, we are changing this system. There are slices of images which we are observing, and we are able to see the tumor and their properties and other things also. So this is what is happening. So this project is also underway. Apart from that, we are working on drug repurposing story, ICMR project, and also that's all. And I'm open for the question and also. Thank you very much. Am I audible? Purva, आवाज आ रही है ना?
Yes, sir.
Yes, sir. Yes. The participants can ask their questions over the chat box. I don't think there are any questions, sir.
So I think then I'll thank you very much for joining in this presentation. If you have any feedback or questions, you can always email us. The email ID is there in the chat box. And if you want to talk to us afterwards, we'll be happy to take your queries and all. Anything else, Sudipti and Dr. Priyanka?
Thank you so much, sir, for such an insightful presentation.
Right. Thank you.
Thank you, sir. I think we'll continue ahead with the second session then.
Yes, sir. So our next session is on transcriptomics data analysis using R by Dr. Priyanka Narad ma'am. Dr. Priyanka Narad ma'am is an assistant professor at the Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida. Her areas of expertise include network analysis, R programming, logic and Bayesian modeling, and machine learning for healthcare and disease management. Dr. Priyanka ma'am also has publication record in high-profile journals such as the Scientific Reports, Interdisciplinary Sciences, Bioinformation, PeerJ, etc. Welcome, ma'am. I would now like to request Dr. Priyanka Narad ma'am to please start the session.
Thank you, Sudipti. Can you hear me?
Yes, ma'am. Your screen is also visible.
Okay. Good evening to all the participants, and I will take you through this session that is on transcriptomics data analysis using R. So, before I start the session, I would just like to reiterate that I had shared yesterday one of the data set, and what I will be covering in my session is a very basic topic that is there. We've been talking about in the last 4 days. I think all the participants and everybody has been hearing a lot on multi-omics and multi-omics data sets and what all you can process and what you can do with the data. So you've been listening on RNA-seq data, you've been listening on bulk sec, chip sec, and all that. But what was not covered so far, I thought I'll just take that portion so that it combines everything and it gets into a complete picture, is the data which is on microarray data analysis. That is one of the things which was not touched so far. So I will take you through microarray data analysis, and I don't know my screen. Yeah. Is it moving, Sudipti?
Yes, ma'am. You just, I think,
It's like,
you were
okay,
full
I have,
screen
yeah,
earlier.
yeah. Yeah, because I can't see, it's not moving when I go full screen. So I think I'll take it like this only. So what I will take you through this session is some of the databases where you will get the data, whether it is RNA-seq data, microarray data, and all the other kinds of data, the public repositories for that. And also I will be processing one of the data sets that are present in a public repository, maybe that you can do for your own research purposes, whatever your research topic is, or if you're working on microarray data analysis, and you can download that data from GEO if it is present in a public repository. And at least the first and foremost step is to process the data. So what you will probably be able to do after this session is at least take one data set from GEO if you follow through along with me, or maybe later, whatever way feels good to you, and process it. By processing, what we mean is I'll show you in the next slides. So let us start understanding about two of the major resources that are there. There are many other resources which you can go ahead and whatever you feel like comfortable with the interface, with the information that is present, and you can go ahead and look for it. So the first resource I'll be talking about and showing you is the NCBI GEO database. GEO is Gene Expression Omnibus. Gene Expression Omnibus is a public functional genomics data repository which follows the Miami. Now, what is Miami? It is a file format kind of a thing. All of you must be familiar about the FASTA file format, but when we talk about expression data, it follows the Miami compliant submission. So if you are thinking about submitting your data to GEO, it has to be present in the form of the Miami or Miriam guidelines, which you call, and you need to follow those guidelines, and then only your data can be submitted to the GEO database. So it has some tools also available. For example, GEO2R tool, which is a publicly available tool. If you are not interested into coding and you still want your data to be giving you at least the differential expression or the values, you can use GEO2R for your basic analysis, and then probably you can understand more about the results that come. So it is an international public repository that archives and freely distributes microarray, next generation sequencing, and other forms of high throughput functional genomics data, which is submitted to by the research community. It also provides a collection of web-based interfaces and applications, which I just talked about, that is GEO2R, where you can download the studies and gene expression patterns can be stored. So if I have to give you a snapshot of what GEO has, it has around 4348 data sets, which is like probably in total, if I have to find about 184107 data sets leading to from those 4348 data sets. And then we have sub data sets, which because there are a broad category which has like maybe 200 samples, and then there are sub series which are available, which has some other samples split into two halves or three halves of the total sample because of submission problems. That is, you cannot upload the whole data set in one go. So you have 24342 platforms, and 5285001 is the total number of samples that are available. So this is a snapshot of a GEO search. What do you see there? There's a search box which you can put this GSC 26787 is known as the accession number, and accession number is a unique identifier that is provided to each submission that happens at NCBI GEO, and it is unique to every record. So you can search either by putting the accession number if you are aware about what is the kind of data that you're looking for, or if you read some kind of papers where you get the information on these data. And then you have the content which is available in the form of data sets, series, platforms, and samples. So platform or GPL is the technology that was used and the features detected. Sample or GSM, preparation and description of the sample. Series defines a set of samples and how they are related. And data sets is the sample data collections assembled by the GEO staff. So all this information, you can see that, and once you click on this, there will be this kind of an image that is generated, which is a full length image of the data that shows you information on the data. So for example, the ID that I have shown here, the status of the ID was public on January 22, 2011. Then the title of the paper, that is comparison of endometrial expression in patients which underwent previous recurrent abortions, implantation failure after IVF ICSI compared to control fertile. So this is the title, and if it seems to you is of interest, you can go ahead and look at the sample. If it doesn't look like that, you are not looking for this kind of data, you can access the information in these terms, and you can then go ahead and work through it. Okay, then comes the organism. There are different kinds of organisms that are available at GEO. There are Homo sapiens, Mus musculus, and so on and so forth. So whatever is your organism of interest, you can go ahead and look forward for that also. Then is the experiment type, that whether it is expression profiling by array, or whether it was a high throughput experiment, or whether it was an miRNA experiment. It will give you the information will be given at this point. Then comes the summary and the overall design of the experiment, followed by the contributors, the citation if the paper has been published for the research work, and the PMID, and the submission date, and the last date of updation. So all this information you will get through the data record that you see in the simple GEO search that you do. So and below this, once you go through the initial methods or information that you want to collect, you can see the platform which is given. Now this is very useful when you will be converting your data. Probes are basically the samples that are present for each gene. There is a probe that is present on the microarray slide. So if you want to know which is the gene corresponding to this probe, you can just use this. You need to know this this platform, which was the platform that was used for the array, that experiment that happened. And then you have the total number of samples. If you click on more there, you will get the whole list. And once you open each of these GSM numbers, you will get more information on each of these records. Then followed by this is the button of analyze with GO2R. What is it? I will just show you in a short while. And after that, we have the download option. That is, you can use the tar of cell file. You can download it, extract it, and you can use it just like I gave you the cell file, which was zipped yesterday. You can download it from here, and then you can run it for your experiment the way we'll do it shortly. So this is what a snapshot of a geo search looks like. Now there are file formats which are present. So there is a series soft files. Soft family files are text files that incorporate complete data and metadata for all platform, sample, and series records in the family. Series mini ML file, which are family files that are XML files that incorporate complete data and metadata for all platforms, sample, and series records in the family. Then there are series matrix files that are text files that include a tab delimited value table generated from the value column of each column headed by sample and series metadata. These files are suitable for loading into a spreadsheet application such as Excel. So this is the different file kind of formats that you saw in the previous slide when I was showing you download family options. So this is what you get there. That is the information regarding these options that you see there. So we go on to each of these file formats. We can you can go to the supplementary file. Now you can have cell files, you can have GPR files. I think it was discussed in the previous session also. Then you can have text files for RNSX, CSV files again for high throughput data. So now the supplementary file contains supplementary files organized according to the entry type, that is platforms, samples, and series. Platform supplementary files are typically related to the array design, what was the method that was used. And sample supplementary files are typically native files representing the raw data or the process data. So depending upon the extension, you can look into it, like suppose it's a cell file or a GPR file, that means it is a raw data file that we are looking at. And if it is having a CHP or bed extension, that means they are process data. And series file would typically include results of upper level analysis such as ANOVA tables or significant G list. For microarray, either the raw data will be present in .cell, .gpr, or .text file format. Today's session, we will focus on .cell, which is the most widely used data type or supported data type for GEO. Most of the cell file data is available with the Affymetrix experiment that are run. For high throughput or RNA-seq data, raw data.sra, which can be converted to FASTQ, and they're also deposited in public database. The most of the times the format is .txt or .tsv or .csv or count matrix file. So let us talk about the retrieval. You can retrieve the data. You can put the GSC ID just like I told you, and you will get the information in the form of the data set that you get in the output, and you can decide what kind of data you are looking at. If you click on HTTP, you can decide the whole data will be downloaded in one go. If you click on custom, you can decide which data you want to process, and you can just get those particular data sets onto your system and process them. Okay, the second database that is also available is all called the Array Express. So now while GEO is hosted by NCBI, Array Express is hosted by EBI. Now, Array Express is also a functional genomics database. It is an archive of functional genomics data that is stored from high throughput functional genomics experiments and provides these data for reuse to the research community. Array Express is one of the major international repositories for functional genomics data, as I just told you, for both microarray and high throughput sequencing studies, many of which are supported by peer-reviewed publications. That is, publication data is also available. Data sets are submitted directly to Array Express and curated by a team of specialist biological curators. Data is collected into MIAME and mini-CQ standards, which are the standards or the file formats that are associated when you click on data submission for Array Express. So by the numbers, it has similar kind of experiment. You can see that submissions through annotate by data type shows that number of sequencing, one color microarray, two color microarray, and plant templates and single sec RNA experiments are present from September. This is just 1 year data that you see on the screen. Now, the search box is similar to that what we have in case of the GEO. You can see if you type in the search box, there will be an option that is given search results for cancer. That is the text box that I wrote here. So you can get the unique experiment ID, which is EMAT TB, just like GEO had GSC number. Here it has EMAT TB and 11300, followed by the curated title, followed by the experiment type, followed by the species investigated, followed by number of assay, and so on and so forth. So now you can go ahead and explore each of these sections. You can filter the search by organism. You can put the organism at Homo sapiens. You can filter the search by assay type. You can click on DNA assay, metabolomic, protein, or RNA assay. You can also create select the kind of platform that you're working on, whether it is Affymetrix experiment that we're trying to figure out, or we can also decide. And you can also, if you don't want to filter, you can just click on cancer and search, and it will give you all the options. So this is again the same result we saw in the previous slide. This has been clicked and shown that how it looks like when you open the data, and you can find more information. Once you work through it, you will be able to understand more on it and understand how the data works in this case. So this is what the whole idea of the databases are there. And before I move on, I would like to take any questions if there are. Sudipti, can you please tell me if there are any questions? Am I audible? Sudipti, you're muted and I can't hear you. Okay. Thank you, Niharika. Thank you. Thank you. All right. So I'll go ahead, and if there are no questions, I'll
Am
go
I
ahead.
visible and audible now?
Yes, you are now.
Sorry, my
And
Zoom was glitching.
that's okay. So we continue further. There are no questions because when I was in full screen mode, I could not see any questions in the chat box. Okay. So I guess we'll move ahead then. Yeah. Okay. Thank you.
So
Okay.
should we move on
So
to the next session, ma'am?
no. I am there. I have a lot to cover. So I'll continue. I just took a break to see if there are any questions. Okay. So can you see my screen again?
Yes, ma'am.
Okay. All right. So if there are no questions on what we just discussed, let us move ahead and talk about the microarray, little bit on the background of microarray before we can go ahead and do some experiments. And again, okay. So microarray is basically a laboratory tool or a slide where you can detect the expression of thousands of genes and proteins. These are microscopic slides that are printed with thousands of tiny spots. These tiny spots contain known DNA sequence or the genes which are related to the probe. So now you generally do these experiments to find out the expression intensities that differ in two different samples. So you have a normal cell, you have a cancer cell, you detect the mRNA, you take out, convert it to cDNA, you label one with green dye, the second one is labeled with the red dye, and then you hybridize the cDNA to the probes on the microarray, and then you scan it through the device. And what you see in the output is what you understand that if it is upregulated or downregulated, or what is the significance of this color. So if you see a gray color or a black color, whatever you understand from the screen, it means that this gene is not present in these cells. If you see a yellow color, that means both green probe and red probe have hybridized, and that gives rise to a yellow color that is present in both cells. If you see a green color, that means it is present only in normal cells, and there is in case of cancer, it gets it gets, the expression level is down regulated or it is not present, and that is why you see only the color that is green onto the slide. And in case of the disease sample, you see a red color only, that is it is present only in the disease cell, or it is present in both cells actually, but the expression is overexpression. So that is why finally on the slide I am able to only see a red color, which denotes it is overexpressed in this sample. So this is a basic workflow where we have the sample selection, then extraction, hybridization, target preparation, microarray scanning, rescaling, adjustment, normalization, standardization, and then you can go on and perform analysis by using clustering, classification, marker selection, etc. So there is a whole lot that happens in the laboratory before it comes on to the system for us. And what you see in the form of cell files are basically these files that have been submitted by after running the experiments. And what we want to do is analyze these files in terms of getting the information through it. So let us go to the hands-on session. That so I would just like a feedback on whether you were able to um, take the data yesterday or not, whether you could take out the data from the chat box. Okay, no, you could not. Yes, so some people say yes. So what I can do is probably upload the data again. And by the time I run it, you can also try to download the data maybe, and you can also run along with me. So I believe I am just uploading it if it works through with you again. So for everybody else, if you are running alongside, I would want you to open your R console, and R console should look like this. So this is again uploaded. So this is a zip file that you see here. What you have to do is download this zip file onto your system. And when you download this zip file, you can save it in the working directory. Now, how do you get the working directory is through your R console. You can just write get wd, which I am writing in the chat box, and you will get the working directory. What I want you to do is download this cell file and paste the data. I am going there. I'm not sharing my screen for now, because when I share, I can't see. Yeah. Can you see my screen now? Yes. Okay. Thank you. So I'll go take you ahead with the whole process. So what I have done is, if you can still see my screen, I have the data with me, the same data that you have with you in the documents, which is my working directory. How I got it, I wrote it in the chat box. So this data is present here. What I have done before I started the session was extracted the data file, and just extract. Once you click here and extract here, you get all the data files that are present here only. So now what you see is this is one file. This is brain1 dot cell, brain2 dot cell, then fetal brain 1 dot cell, fetal brain 2 dot cell, fetal liver 1 dot cell, fetal liver 2 dot cell, liver 1 dot cell, and liver 2 dot cell. So all these cell file extensions will be extracted into your working directory like this, how you see in my system. So if you are able to do that, please tell me. Please write a yes if you are able to just download and extract it on your screen. Please tell me, or should I just tell you the further steps? I hope this much is clear, because this is the starting point that we do. If you have to, how you get this data is, I told you, through GEO. If I have to take you through GEO, I will just say, like, you can just type GEO data sets and you can type the data here, the number, whatever number you are looking for. So let's say I am looking for GSC 21222. That is what is the data I am looking for. So once you click on search, you will get that data onto your screen. And yeah, from here also, when you click on HTTP, you will get the same kind of files which I have already provided you, because the it takes a lot of time for downloading. So that is why I had downloaded it for you and provided you the cell files. Okay. Okay, great. So if we are there, let us start the process. So the first thing we do is we come to our R console. So I had told you to download this file, that is download R 4.2.1 for if you're working on Windows or Mac or whatever you are working on. You just have to download R on your system. The first thing we do is download Bioconductor or through the BioC manager. Now, Bioconductor is also a public repository which has a lot of information that is present. It has a list of packages which are useful for genomics research. You will get a lot of packages for different purposes, for annotation, for processing, for analyzing. Everything is available at the Bioconductor platform. So the first thing you write is this. I will paste this also in the chat for you to follow along. So if you can just type this, you will have to put it into the R version. So once you click on enter, it will take you through this, and then you will, through the BioC manager, you can just extract install the whole Bioconductor list of packages. It will assign, because for me I have been installing it n number of times, so it is already there, and it is just updating the packages. First time when you do that, you will be having some packages will be updated, which you can do easily. Okay, so this is what you write in the first go, and you can see that it will run. And once it runs, you will see that it gets down to installing all the packages that are available. And if you have used it before, it will only update the packages that are new or that have a new version. And it will ask you update all, some, or none. You can just write A, that refers to all, and all the packages will be installed. So the second thing I need for my analysis is the FE package. That is, I need this to work through. So I will write BioC manager and install, and this is how you install any package individually. That is, you just write in brackets, you can just write the name of the package that you are looking for, and you will get the information on that particular package that is installed. So followed by this, you can call the library. That is the second step you do. Once you install the package, the next thing you do is you call the library. So once you are through that, you can just write library FE and try to call the library that is there. If you are running everything correctly, you will not get any error up till this step, and it will keep on updating itself and loading the required packages that are needed. So once you are through this step, you need to write and call the Ephemetrics data. So Ephemetrics data can be called by writing this line, that is fe.data, or this is just a name of the variable. You can write anything. The function is readFE. So R is a case sensitive language. You have to be careful by how you write it onto the console. So you need to write it like R capital EAD, A capital FFY, and empty brackets. So whatever cell files are present, they can be 1, they can be 10. Everything will be read by this package, which is present in your working directory. So I repeat, how you can get your working directories just by writing get WD. It will give you a path, which is your working directory. So from that working directory, if the data is present there, it will be automatically read onto the system. So this is how you can read the files. That's the first thing you do. Once you do that, you can just use this command. So now why am I using eset.mas5 here? Because this is what is the processing scheme I'm following, that is the mas5 algorithm. There are several algorithms other than this, that is it is mas5 RMA, it is you can use GCRMA. These are different kinds of background correction algorithms that are present, which you can use according to the experiment type. mas5 is widely used, and so is RMA. So any of these will work. So if you're using RMA, you can just write the name of the variable eset, that is the variable that I'm using in this case, dot RMA is equal to RMA, and then the data you want to process, which is the name of the same variable that you used in the previous section. So you will write this and process. And once you do that, you will see it will take a little time to process the data. Then since there was no log values that is present, so I'm going to use convert it into a variable, where I'm using the variable experset and no logs, where I'm putting the expression value into of eset.mas5 into this variable that I have created, which is a no log variable. And then when I click this, I put this, the next thing I do is convert or use a log transformation, which is an excellent method for working through the normalization of the data. That is, I can put it experset.no logs, that is the same variable I used in the previous step, and comma 2 means log to the base 2. I'm running a simple normalization where I'm converting all log values to the base 2, and this is what I can do. Finally, what I want to see is the expression value. So I want the expression values. I can simply use the command write dot table, and I can just give the format to be here. I can write dot table. The value experset is this variable, so you have to put the variable where you have log transformed your data. File is equal to su mas5 matrix 2. You can give any name you feel like. .txt is the extension. Quote is equal to false. I don't want any quotes in the data, and separated by tab T means tab delimited file. So once you run this, you can see if you have not, there is no error in your system, and you can get the final file that will be present, which will be a processed file. So your processed file looks like this. If you see, this is how the processed file will look like. I don't know whether it is visible on your end. Can you see the text file? Or are we still on the R console?
We're still on the R console, I think.
Okay, I think it's a little slow. Yes, so Nitya can see, but I can't see. Uh, should I share again?
Yes, ma'am. I still can't see the file. I can
You
still
can't
see
see
the
the.
R console.
Okay, let us try again. Mm, okay. Can you see now? Okay,
Yes, ma'am. Yes, ma'am.
yeah, okay, so okay, great. So now this is what you will get once you pre-process your data. This is the name of the samples that you see here that I had shown you in the beginning: brain 1, brain 2, fetal brain 1, brittle brain 2, liver dot cell, and so on and so forth. And these are the probe IDs. Every probe is written here. These are the expression values. So the first probe that is there, the expression in brain 1 dot cell corresponds to this, then the brain 2 dot cell, because this is a replicate experiment. So you can see the expression values are somewhat similar. Then the fetal brain has a lower value, which is same in fetal brain 1 and fetal brain 2. You can see there. And then fetal liver has a different expression value, and you can see that the expression values in fetal liver are lower, and whereas the adult liver cells have a higher values that are present here. So this is just a snapshot of what the experiment looks like for every gene, and this you cannot see unless you process your data through R. The dot cell file will not open directly. You need to process it using R console, or you need to work through R to understand the data or to work through further. You can go ahead and look for these probe. There are probe converters. You can use gprofiler also for the same. And this is going to help you with the understanding of which gene this is, because there will be multiple probes that correspond to the same gene. So if you see, it is a huge data set, and once you convert it to these FE are the base housekeeping genes. Other than that, all the genes that are present for this particular array, for this experiment that we have run, you will see that those values are present here. So this is how you process the data. And if you want to run along, or if you want to if you have any queries, I can give you 5 minutes or maybe 10 minutes, and you can just check if you can run the code or not. Or if there are any queries, then I would take them, and then we can proceed further on the next thing that is there. So okay, are there any questions? Yes, no? I have put the whole summary of the code in the chat box for those who are working through it. If you really want to work through it simultaneously, please do that. And do let me know if you face any troubles. So in the next 10 minutes, I'm giving you 10 minutes time to run it, and then I will show you a tool that will perform the differential expression for the data, that is GEO2R, which is a 5 minutes demo. How can you run through it, and how is it visible? Okay, so okay, then I can stop sharing for the next 5 minutes, maybe 5 or 10 minutes. Please provide the GEO identifier. Okay, so you can work through any data. This is a downloaded data that I had with me. You can use the data set that I gave you in my slide, which is the I'll give you the data name of the data. So 26787 GSC 21222 is what, if you want to go through GEO and run it, so you can use GSC 21222 and run it. You can download it, but downloading simultaneously will definitely take a lot of time. So that is the reason I had given you the data, and you can run it probably and check if it works for you, because this is not the complete data set that I am using, because otherwise it would have not run in this time. So that is why there is no fixed identifier for this data that I have right now with me. These are some part of the sample. So if the sample had replicates that were like 30 or 32 for each sample data, like for fetal brain, I am using only 2 at a given point of time, just for the ease. So I have not downloaded the complete data set. I've downloaded using the custom option, so that you can process it in this time frame of like whatever 45 minutes or 1 hour I had with me. Okay, so all right, I give you 5 minutes. All right, so I don't see any questions in the chat box. So I am not sure whether you are running alongside with me or you are going to try it a little later. So in any case, if you have any queries, you can reach out to me. And what I'll do is I'll take you through another part of processing the data, which is through an online tool, which you might have or might not have used so far. So what I'll do is I will take you through this tool now. Okay, so, and I'm going to share this handout with you, which you can try working through later with the same data set that is provided to you. Now, this handout has all the information of how you can pre-process and normalize FU matrix expression data, the commands that you can write, followed by the description to each command. What do we do? And then some optional step, which I just told you that in case of you can use Mass5 and RMA or GCRMA and so on and so forth. You can also normalize the expression value which is present with the matrix file that you have just created. You can use that, and then you can again find out the trimmed mean or normalize the data. You can also perform log2 ratios. You can combine all the data together. You can perform t-test on the data with the same file that you have just created, and then you can do all these commands one by one. If you run them correctly, before running them, of course, you need to install bioconductor onto your system through the BioC manager, which I told you in the initial steps. And then you can perform t-test, you can find out the differentially expressed genes, and so on and so forth. So this whole handout I will also be uploading at the end of my session. In case you want to work through more on this, you can just try it, and if you have any troubles, you can reach out to me anytime. So now what I also wanted to discuss was one of the another packages that is present there, which is not package, actually it is a tool which is present. So here I have searched for GSC 21222. This is what I am looking forward to. You can put it in the chat box, and once you put it in the search box, what you get is a page like this. So now this is a description. I already showed you in my presentation what each of these look like and what are they referring to. This is the summary of the experiment. So this is basically an experiment that was done to compare human embryonic stem cells with biological and epigenetic characteristics similar to those of mouse ESCs. So if I want to, there are two conditions in stem cells, naive and primed, that is the two conditions. If I want to compare these two conditions and have a better understanding on them, I can see that there are 18 samples in total. The ones that are starting from 13 to 18 are the naive samples, and the ones that are before this are the prime samples. Most of the ones are the prime samples. So when I am going to compare the data in GEO2R, what I need to know is the differentiation in the samples that I am looking for. For that, I need to know which is the naive sample and which is the prime sample. Now, in this case, it is clear with this heading. Sometimes there is no description, like for this case, there is no description. So if you want to know what is it about this data set, you can just open the whole link, and then you will get to know, okay, it is a naive sample that is present. And you can decide upon the description of the data just by clicking the link into a new window. So you can do that, and you will get more information on which is the prime and which is the naive, or whatever the condition it can be a naive prime, or it can be even simple normal and diseased. If you're looking for some disease samples, whether it is cancer or something, you can just look for normal samples and disease samples simply. So now below this, there is a button which says analyze with GEO2R. If you are not into coding and you do not want to go ahead and go for the processing using R, and you are not familiar with R, what you can do is click on analyze with GEO2R. And once you click that, what will open is a page where you will see, just a minute. Okay, so when you click on this analyze with GEO2R, it will load into a screen like this, where it will give you the accession number and the summary of the data that you have. Now, the first thing you have to do is define the groups. So I will define two groups. I will define one group as naive and the other group as prime. Okay, you can do that for your own experiment, and then you can put the data into them. So how do you put the data into these samples? You just have to select the ones that you want to put together. So for example, I can just see and know that these three are let us combine the ones which are HESC and do into HESC and HISC. So I'm going to put all this data into knife, and I'm going to put all this HIPSC data into prime. So you just control and select, and then you can put them all into this prime. So 4 samples I have put to knife and 4 samples I have put to prime. Whatever you want to send to the particular group, you can just click and send, click and send. That is exactly what you need to do. This is very, very simple. And then once you are through your selection, what you can do is just click on analyze. Okay, so now when you click on analyze, it will process the data for you. It will do all the work that we were just doing a few seconds below for yourself, and it will take a little time, which is what it should be with the amount of internet that you have at your place. It should be done in the next 5 minutes. So it is going to give you the options, that is going to give you a graphical results, that is going to give you the values, the metadata. Yes, now you can see that the data is there on the screen. So what you see is the top differentially expressed genes. You can download the table, full table. You can select columns that you want. If you want to check columns that you don't want these values, it's fine, you can do that. Or you can just go back and download the whole table. Now the first is the ID, followed the IDs, the same IDs that you were seeing when you were processing the data, followed by a statistic that is adjusted p-value, t-value, b-value, and log FC value. So these are all statistics which we can also generate if we are using the lemma package in R. We can generate this and we can get the value. This is the gene symbol, which is that it has done extra to your work. That is, according to the probe ID, it is giving you which gene are we talking about and the gene title. If I have clicked on this gene symbol, I am getting the expression value of the individual in naive. That is, I can see very clearly that it is showing expression in naive, whereas the expression is very minimal or not there in prime. So this is an excellent thing that I can just visualize and tell, okay, this gene is a differentially expressed genes because it is only expressed in naive and not expressed in prime. Likewise, this is not having any gene ID, so it might be a duplicate gene that is present. If I click on the second one, again, I can see, okay, great, naive versus prime. It has expression value in naive and not in prime. And I also have the gene title. If I want to get more information, I can go to other databases and get the information. Now, these are a list of visualizations that have been created. This is the graph that you see is, okay, log2 fold change against the p-value against fold chain, and the ones that are upregulated and downregulated. The downregulated ones are in blue, the upregulated ones are in red, and so on and so forth. You can explore. This is a volcano plot. This is basically known as a volcano plot. So you can explore more towards them. So then you have the second kind of plot, which is also there, which is a mean difference plot, which will give you the same kind of information in a different output, and so on. Box plot, intensity plot, so on. Whatever is significant to you, you can just pick that. And if you want to change any option, you can click on reanalyze. And when you click on reanalyze, it will tell you, okay, this is what we were running, and if you want to change anything, you can do that by clicking on reanalyze. The default settings can be changed, and according to your experiment that you want to run, you can use the data set by that manner. So let's see if it loads. Yes, so I can see here again, reanalyze was giving me the same data with the same kind of options. Now if I want to change the options, if I don't want this kind of p-value statistic that is applied, you can change it to Hochberg. These are different kinds, Bonferroni. These are different kinds of methods that are applied for finding out the p-values. If you want to apply log transformation, you can say yes. You want to have lemma precision, so that is what I was telling you. We can also do the same by actually giving the same in R using the lemma package. Force normalization, if you want, it's fine. You can click yes, no. Platform and reputation, that you are seeing the gene name. we are saying that we want to go ahead with the NCBI annotations and so on and so forth. So you can be generating data like this. Then profile graph will give you the ID if you want to set that. This is the R script that was running behind what you saw, and you can actually do this a little simpler, in a simpler manner, by just running the R code yourself. But if you want to check what is happening behind this data, so you can just simply check the R script for it and understand what is happening with the data set that you just used. So in this case, I used only 8 out of 18 samples. You can go ahead and analyze more information. You can add groups. I've only added 2 groups. You want to add one more group, suppose you have a control, you have a normal, you have a disease. If you want to run between the control, the treated, the normal, you can just create as many samples as you want, and then you can select what do you want to match. When you click on options, it will give you up to 5 values, and you can just go there and select, okay, I want this, I want the contrast plots for all these samples. So this is what you do, and this is how you can process the data. If you are not interested in working through R, there is another way of doing that. Many publications use GO2R also as a method. If you want to follow it more, there's a YouTube video, which is a tutorial video, which will just tell you the same things I just told you, and full instruction will give you the file will give you the information in a manner which will be easy for you to understand. So that is all that I had to discuss with you about the data that was not discussed in these 4 days, that is the microarray data analysis. GO2R can be run with RNA-seq data also. You just have to see for that option. If any data has it written there, analyze with GO2R, that means it is compatible with GO2R, and you can go ahead and analyze the data for yourself. So that is all from my side. If there are any questions, I would be happy to take them. If not, you can connect with me anytime you feel like, and we can discuss more on what you want to do with your work, how you are interested to look forward to it. So I guess
There are no questions in the chat box.
there are no questions. So I believe everything is clear to everyone, or probably they'll work it a little later. So I'll share the tutorial with you. You can go ahead and work through it whenever you feel like, or whenever you want to work on it. Okay, great. All right, so I think I'll close. I am already 1 hour into it, I think. Yes, okay. So okay, that's all from my side. I will put the data set. I think we can go ahead with the next talk, or maybe we can take a 5-minute break.
We can start at
And then
5:50, ma'am.
we can start at 5:50. Okay. By that time, I'll paste the links and the feedback link also for today. So we can start at 5:50. All right. So, okay.
Thank you so much, ma'am, for such an insightful session.
Thank you, Sudipti. I think everyone can take a short break. We start at 5:50 again.
Ma'am, should we start?
Yes, am I audible?
Yes, sir, you're audible. So our next session is on metabolomics analysis by Dr. Abhishek Sengupta, sir, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida. His research goal is broadly focused on the development and application of predictive computational models and associated web tools or databases for predicting and analyzing the behaviors of complex physiological systems ranging over multiple scales of organization, from the molecular level to the tissue, organ, organism, or the population level. I would now like to request Dr. Abhishek Sengupta, sir, to please start the session. Welcome, sir.
Is my screen visible?
Yes, sir, it's visible.
Okay. So I'm Dr. Abhishek Sengupta. So by now, I guess everybody's very tired, exhausted with all these days uh, sequence sessions in the evening. People were working in the morning and then attending this workshop in the evening. So I am going to speak about or talk about that genre of omics, which is little different from what you have been studying all these days, right? So this omics, that is metabolomics. My topic is metabolomics. So this omics is uh, has some specialty. I'm going to tell you about that. That why this omics is just one second. Okay. So this metabolomics is that type of omics where we study about metabolism, right? So metabolism is something which is happening in your body all the time, happening in all organisms all the time, whether it is whether it is in a healthy condition of a cell or unhealthy condition of a cell. Unhealthy condition means disease condition, or probably some fluctuation that must have gone through the cell, right, or through the organism. So metabolomics is that type of profiling. So these days you have been studying about data analytics and profiling of genetic material, uh, genome, your proteome, your transcripts, single cell, RNA, DNA, fine. So what we have missed in this whole workshop is the third category, that is proteomics, and the fourth category, that is metabolomics. Though we have missed proteomics, but I will tell you there is one similarity between proteomics. We will not miss it because the techniques that we use in profiling a proteome or a metabolome are almost same. Fine. So the techniques that I will tell you are more or less same in case of proteomics also. Though the data size or data in the language of data science, it is little different because the size of the data, the texture of the data, so that varies in case of proteome and a metabolome. Now let's understand the basics of metabolomics, because it's an evolving field of omics, metabolomics. Metabolism you have studied a lot. You have studied the biochemistry pathways, biochemical pathways, etc. But metabolomics is something which integrates all the metabolic pathways in an organism, right? So how these pathways integrate among themselves, this is what is metabolism. So now next I will show you one picture. So you must have seen this picture. Uh, Probably, I don't know whether you have seen this picture. It looks like a very complicated circuit, but this is, believe me, this is not a circuit. This is actually the metabolic, whole metabolic network of human. Fine. So you can see here the carbohydrate metabolism. You can see here lipid metabolism. You can see here amino acid metabolism. So it is actually the overall metabolic metabolism in a in an organism in the form of an integrated network of metabolism or metabolic network. Fine. So metabolomics is all about studying or profiling or identifying these these different aspects of these pathways. Now, what are these different aspects? These different aspects are the metabolites, are the different energy currencies, different cations, anions, right? Water molecules, the different uh, uh, the hydrogen atoms, like carbon dioxide. So all these form uh, uh, ca- um, the vitamins, the hormones. So all these are basically the composition of a metabolome, fine. And how this composition uh, is important is important from the perspective that uh, that genotype, studying the genotype or studying the genome, studying the transcriptome tells you a story, tells you about or predict give you prediction about various biomarkers, gives you the prediction about various target identifications, gives you idea uh, about the disease or finding out various uh, or understanding the molecular mechanism. But this one omics, that is metabolomics, is the bridge between the integral genome and the phenome. What is phenome? Now, this is another very big word that you are, I think, coming across. Phenome. Phenome is nothing but the whole overall phenotype of an organism is called as phenome, right? So there are many ohms or omics. So out of them, there is another one that is called as phenome, that is phenotype. So this metabolomic is the bridge between the genotype and the phenotype. Fine. So this is so the one of the most important uh, advantage of studying metabolomics or metabolic profiling work or understanding metabolomic data is that it tells you directly about the condition of the disease or condition of phenotypic condition or the physiological condition of a particular cell or a particular tissue or a particular organism. Fine. So this is what we see in metabolomics. Now, the metabolome. So, what is the metabolome? So, you can see here the genome, right? From genome, the same central dogma of molecular biology, like from genes to mRNAs to proteins to metabolites. Now, here we have added one more thing, that is metabolites. From proteins to metabolites. What are these metabolites? What are these metabolites? Because metabolomics is nothing but the overall composition of all the metabolites that are present in all the metabolites and the metabolic pathways that are present in a cell, in a tissue, in an organism. Fine. So the metabolome is the complete set of metabolites within a cell, tissue, or biological sample at any given time point. Now, this is called as time series metabolomics. This is something very, very high throughput. Time series metabolomics, where we see the concentration of the different metabolites at different conditions, right? Or at different time intervals, right? So this is called as time series metabolome. Fine, but the metabolome is the so this is very important to mention that at any given time point, what is the concentration of a particular metabolite? So metabolome is inherently it's basically around the metabolite size. So you must be understanding which we are considering as metabolites, like the carbohydrates, lipids, amino acids. These are all small molecules, right? These are all small molecules which are less than 1500 kilodalton. So that is considered as a metabolite. So they are uh, found directly in your cells, in your blood, in your blood, like not the biofluids, not only blood, even like sebum, saliva, like different swabs, like urine, tissues, fecal samples, uh, tissues, organisms, etc. Organisms means microorganisms. So, so these is the uh, type of uh, experimentation that we do is non-invasive nature of metabolomics, and its close link to the phenotype make it an ideal tool for the pharmaceutical today in pharmaceutical, preventive healthcare, and agricultural industries. Among it's one of the uh, what should I say, buzz omics these days, which is very much useful. Like, for example, the reports that you see, the various diagnostic reports that you see, right? So what are they? They are nothing but they are the different metabolite concentrations or the different metabolites at certain point of time, either at the fasting stage or at the feeding stage or at the during exercise condition, or a heart patient or a cancer patient, or like no any phenotype. It could be any phenotype. So these metabolites tell you the exact the exact exact story about what is the status of uh, of your condition. For example, I'll tell you one theory, that is Warburg effect in studying metabolism in cancer, where they have seen that because in cancer cells there might, because of the absence of mitochondria, the glucose enters, but it does not produces pyruvate, it produces lactate. So the overproduction of lactate has shown that yes, the cell is cancerous. It has been observed in breast cancer. So it has been studied in breast cancer and also in other cancers also. This Warburg effect is seen, and it is being studied, right? So biomarker, so in biomarker discovery, it has a huge application. Drug safety, in toxicity analysis, in toxicology, it has a huge application. In metabolic engineering, it has huge application. In biotechnological applications, it has huge application. Fine. So metabolomics is something where we quantify, where we study the uh, quantified metabolites, right? The metabolites that has been quantified through various uh, experimental techniques. Fine. So in personalized treatment also, it has a huge role, personalized and preventive medicine. Now the omics science, so if we see side by side from genomics to transcriptomics, so you can see here in genomics we deal with around 40,000 genes. In transcriptomics, the number has increased, the data size has increased to 1,50,000 transcripts. Now in proteomics, the data size is almost more or less same as transcriptomics, that is transcript, that is 1 lakh proteins. But if you see here, in metabolomics, the size of the number of metabolites is very low. So our data size is not huge, but it tells you an exact story about what is the present status or condition of a particular uh, cell or a tissue, fine, at a particular period of time, right? Now here you can see the techniques that are used in case of, just ma'am has taught you microarray data, that is microarray data or SNP array SNP. There is in transcriptomics we have RNA-seq data sets and single cell, bulk RNA-seq, etc. Microarrays also can be considered. So in proteomics we use in proteomics and metabolomics we use almost the same technique, same bioanalytical technique to generate the data. That is uh, for example, MALDI-TOF, MS, LCMS, ESI, MFMS. So these are also same in case of metabolomics also, along with NMR. So here, so these are the various data outputs. So you can see here the data outputs in case of genomics, transcriptomics, proteomics, and and so in proteomics and metabolomics, for example, if we take the MS, that is mass spectrometric data, so we get peaks. Now, so I will come to that later on when I will talk about the case study. You see here the same thing that I have just mentioned, that 25,000 genes, 7,500 enzymes, and 8,000 chemicals. So this is a pyramid which shows you the number of components or number of let's know the data size that keeps on decreasing along the pyramid if we go from bottom to top, right? So this is genomics, this is proteomics, this is metabolomics, fine. So, so all these comprises, so this multi-omics data analysis is nothing but what we study in systems biology, fine. Integration, multi-omics integration. So metabolomics, so if you study a certain disease, so if you integrate metabolomic data as well, it gives you a clear picture of what is the status of that particular disease, or what pathways, what enzymes, what substrates that are participative or that are expressed or highly expressed um, in, uh, in a particular condition or in a particular disease condition. So that can be integrated with the genomic and transcriptomic uh, data to get more uh, truthful results. Now next is traditional metabolite analysis that we do. We use this HPLC, gas chromatography, mass spectrometry, right? So what we get as the data from these techniques are the peaks. So these are the different peaks that we get. So measure multiple of metabol. So it measure multiple of metabolites at once, no separation. So it allows metabolic profiles or fingerprints to be generated. Mostly, they are mostly automated. Relatively little sample preparation or derivatization that happens. So it can be quantitative, and we can analyze it very. So what is the difference between MS and NMR? So we can use both these techniques in generating metabolomic data. In case of MS, is that the coverage is very high. So we can cover a lot of, we get a massive number of metabolite information, but the quantification is not that perfect in case of MS. right? So that is much better in NMR studies, right? So in NMR studies, the quantification is quite accurate. So there are two routes. One is targeted. So these are all classical metabolomics, right? So one is two approaches are there. One is targeted profiling, and another one is chemometric or non-targeted profiling. So targeted profiling is a bottom-up approach, and chemometric is a top-down approach. In targeted profiling, the compounds are identified and quantified prior to analysis. So that's very much obvious from the term itself that targeted profiling is that you want to target a particular cell type, or you want to target a particular type of pathway that you are targeting. So that is targeted profiling. Untargeted profile, non-targeted profiling or chemometric profiling is that you have the sample with you and you identify different patterns. So they both have their own use, but the most useful thing in metabolomic data science is the targeted profiling, right? Even non-targeted is also used. Targeted is more useful, but non-targeted is massively used, right? Because of its ease, right? The ease of doing it. Fine. So next is again this untargeted metabolomics is a comprehensive analysis of all the measurable analytes in a sample, including chemical unknowns. So due to its comprehensive nature, untargeted metabolomics must be coupled to advanced chemometric techniques such as multivariate analysis, univariate analysis. No, but in this case, multivariate analysis to reduce the extensive data sets generated into a smaller set of manageable signals. Right, so it generates a huge scale of data. So to reduce that, we do the multivariate analysis to get manageable signals. So these signals then require annotation using either in silico libraries or experimental investigations, and subsequent identification using analytical chemistry, right? Untargeted analysis, untargeted and untargeted analysis is more useful technique as compared to the targeted approach. Fine. So let's go to the next slide. So now there is one aspect that again I will say that I already said that metabolomics is the bridge between what genotype and phenotype, right? Now there is actually a bridge that also acts between metabolomics and phenotype, and that is called as fluxomics. Fine. So the fluxes that like the flux is what? Flux is basically the flow of metabolites, the rate at which the flow of metabolites through the various pathways. So there are various fluxes at various points. So there are huge number of fluxes as there are huge number of pathways, right? Or reactions, biochemical reactions. So every reaction has a particular flux, right? So that distribution is called as flux distribution, and studying of flux distribution is called as fluxomics, right? So metabolomics and fluxomics are very closely related to each other, right? So DNA, RNA, protein, metabolites. So ultimately, what information you get from DNA is what is possible, and RNA gives you the answer of what appears to be happening, and proteins give you the signal that what makes it happen, And metabolites actually tells you what is happening, right? What exactly is happening. So did you get did you understand this picture, that DNA where what tells what? Like DNA tells you what is possible in a cell. Like what could be the possibility? RNA tells you what appears to be happening. Proteins tell you what makes it happen, and metabolites tell you the what is happening actually. Fine, metabolites. So metabolic flux analysis, I am skipping this slide. So the metabolomics has a big role or huge role in metabolic engineering, right? Metabolic engineering. So metabolic engineering. So they have metabolomics has many applications. The first one of the age-old application of metabolomics is in the field of metabolic engineering. Now, what is metabolic engineering? It aims, so we increase the production of or we optimize the production of certain metabolites, right? Suppose we wanted to optimize the production of a certain antibiotic in from a microorganism. Now, antibiotics are what? Antibiotics are secondary metabolites, right? The antibiotics are secondary metabolites. So, if we wanted to increase the production of that particular secondary metabolite in an organism, we need to engineer that organism. So here comes your synthetic biology and genetic engineering aspects, right? But we can do it computationally also. We can engineer the system in such a way that the cell or the organism produces that particular optimum or do that optimum production of the desired metabolite. Fine. For example, penicillin, for example, various alkaloids on various these secondary metabolites used in nutraceutical industry or biofuel industry, etc. Fine. So first, artificial metabolic pathways are designed. So what happens? What we do computationally? What is computational biology or computational systems biology? So here systems biology we use. So second, protein evolution is performed to construct and optimize the design pathways, and then metabolic flux is driven to the production of target compounds. Now, metabolic engineering is a what it's an continuation with the previous slide. So, metabolic engineering it defines the modification of specific biochemical reactions that are happening in an organism or that are present in an organism to produce the required amount of the desired metabolite through uh, recombinant DNA technology or RDT. So this happens in actual environment, right? But you can also mimic it computationally by studying the generating the metabolic networks or and then ultimately running the simulation or uh, 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 these the reactions by running the reactions computationally. So considering its advantages over the other chemical synthesis routes, this area of the biotechnology is likely to revolutionize is fine. So once biochemical reactions targets have been identified, established molecular biology techniques are applied in order to amplify sorry in order to amplify the in order to amplify uh, the metabolites, fine, different or the enzymes or their corresponding enzymes. So it's a four-step process. Basically, this metabolic engineering is a four-step process: design, build, test, learn. So I'm not getting into that part. So that is more of like metabolic engineering class. So metabolomics workflow. So this is a typical experimental workflow of metabolomics. Like you have a biological question, experimental design, sampling and sample preparation. So you do the take the samples. Suppose you take the blood sample from a patient. You do the so the directly the blood sample can be uh, taken and analyzed in the mass spectrometer or MALDI-TOF or NMR. So that is data acquisition and then data pre-processing. So the next part is rest of the part after these peaks are generated, that is all data analytics. So that is called as metabolomics data analytics, right? Metabolomics data analytics. So we use various techniques. We use traditional ways, traditional software tools. Also, we use ML or AI-based techniques. So what happens in metabolomics data? So before that, let me just complete this part. So we take the sample type like cells, tissues, right? This is again in continuation of the previous slide. So it's almost like a repetition. So then data acquisition, metabolites are isolated from the samples and data is frequently acquired by a first step of separation with liquid chromatography before injection and analysis in a mass spectrometer. So that is all about LCMS. Uh, Bioinformatic tools are necessary to pre-process LCMS data, which involves extraction of metabolite features and their quantification. Fine. Metabolite features and their quantification. A single metabolite can be represented by multiple peaks. Annotation can overlap with pre-processing steps or be performed after downstream analysis. Statistical analysis, enrichment in metabolic pathways or pathway enrichment, and integration with other omics data are carried out to gain insight into this. I have already mentioned to you that these metabolomic studies along with other omics data. So that is all about multi-omics. I'm finishing this workshop again with the same theme that it's all about multi-omics analysis in systems biology, right? You use different omics data, the more insights you will, the more much better insights you will get from the data. Fine. Next is there are two types of data that we get in metabolomic experiments. Metabolomics experiments, right? So one is these peaks that we get, right? The peaks spectra, or we call it as spectra. And the other type of data that we get are MZ data, MZ values, right? That is mass charge values, right? So either they are in the form of peaks, right? Or they are in the form of, in an Excel sheet, they are in the form of these mass charge ratios or values. So you do any if you, so for example, there are geo database, there are bio project databases. So all these databases are for various various data sets. So for metabolomic data sets or data sets, metabolomic experiment data sets, we use different databases. I'll come to that. I'll show you some of the databases. Just the list I will show you. I'm not like getting into the any of the particular databases. I'll just mention some of the databases. Like there is this metabolomics workbench by NIH, and then there is metabolites by EBI. There is MetClean database, right? So all these databases will give you the data sets. But the experiments, uh, uh, getting too much data is not feasible, because people are doing it gradually. People are submitting data, as compared to transcriptomics and genomics. These data sets are very rarely available. You have to ask, because they are not documenting properly, and even the databases are not very, even the HMDB database is also there. So these databases are not very well established. So they're well established, but data submission rate is very less, right? Because of the expense of, or because of this instrumentation for this for generating this data is it's little high. So because of that, like for example, sequencing, like these days there are so many types of sequencing has come, but there are no alternatives of mass spectrometry and NMR. We are still dependent on MS and NMR, because they are the gold standard methods by which generate we can generate metabolomics data. So there are various applications of metabolomics. So in plants, microbes, animals, in every sphere of life, these metabolomics can be applied. Biofluids, you can use biofluids. You can use cellular cell cultures, right? Detection of biomarkers of diseases or drug diet related changes. Metabolome is the closest representation. Then toxicology testing, clinical trial testing, food and beverage tests, nutraceutical analysis, genetic disease tests, drug compliance, transplant monitoring. So huge applications metabolomics has, and it has a huge prospect. Application of biomarker identification. So what we people are doing these days are mostly like biomarker identification. So you take the biofluids from human, you do mass NMR or mass spec or some chromatographic techniques, or coupled chromatography and mass spec. Then we do so these profiles are generated in the form of spectrums, and then we do PCA, PLS analysis, discriminant analysis, all these statistical based analysis. And then we search the database. For this we do the pathway enrichment analysis also, and then identify the biomarker validation, that in which pathways these biomarkers are present. Like, so these are the various techniques by which we can generate mass spec data. So this is one project. I think I should mention this project by University of Alberta in Canada. That is the Human Metabolome Project, like Human Genome Project. So there is a very highly funded project that is that 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 is still ongoing project. People are doing a lot number of uh, additions to it. So that is though Human Metabolome Project is considered to be completed, but still there are some updations that are happening. So So there are other metabolomes also, like yeast metabolome, then E. coli metabolome, because these are the model organisms, because these are more needed for metabolic engineering. That's why this E. coli and yeast metabolome has been sequenced, or sorry, not sequenced, has been uh, profiled. So then, sorry, characterized. Then HMDB, that is Human Metabolome Database. This is a part of the HMP project, that is Human Metabolome Database. So you can find almost a huge amount of data from Human Metabolome Database about the metabolite concentrations in cancer, in healthy conditions, but you don't get the data sets. You don't get data sets in HMDB. You get only the metabolite information, but you get data set information, data sets from the databases that I have mentioned, that is metabolites. uh, then metabolites means METABOLIGHT, metabolites, metabolomics workbench, metlin, etc. Right? Human metabolome project, I have already mentioned about it, right? So this is the amount of data. So human metabolome project has many tangents. So they have also worked on the various toxins that are present in that affects the human, the drug metabolites, food additives, phytochemicals, drug. They have been linked to drug bank also. This human metabolomic database is uh, like almost around 8,500 metabolites that have been sampled in HMDB database. Right, so these are more applications. Why is human metabolomics important? Because it is useful in diagnostics. It is to identify the known drugs are small molecules, because most of the drugs are small molecules. So they come under the category of metabolomics most of the time. So you can use a lot of different ML based techniques also to uh, to process them. So of all drugs, 50% of all drugs are derived from pre-existing metabolites. 30% of the identified genetic disorders involve diseases of small molecule metabolism. Right, small molecules serve as cofactors and signaling molecules to thousands of proteins. So this is the importance of small molecules, and wherever there is importance of small molecules, that is metabolomics. Types of search queries supported by HMDB. I think I'm keeping this. So this is about HMDB database. What are the different types of information that you can search, like structure search, text, molecular weight search, substructure search, smiles and formula search, um, then MS spectra search. So all these informations are integrated in HMDB database. Fine. So for modeling, for various pathway-based modeling or metabolic network modeling. So that is my area basically. I do research. Our lab also do research in metabolic network analysis or modeling and simulation. So we use the Copacy, Cell Designer, Accelerator, Cobra Toolbox, and stuff, and Raven, etc. So that's all about metabolic modeling, where we model the metabolic pathways. Fine, and we run various analysis. One of the most important analysis is flux balance analysis. There are other analysis like MOMA, room, etc., FVA, metabolic analysis. So I think I have spoken about it. These are some of the databases. It's very small. I think you can take a picture of this and little magnify it and see it. Human metabolome database. Then there is KEGG. KEGG database is also quite useful in metabolic network research. Metapsych, human psych, right? So CHEBI, uh, Metlin. So all these are different. NIST. NIST is also quite useful. NIST, but NIST is mostly useful for protein spectra, basically for proteome space, proteome analysis. These are tools for metabolomic interpretation. So there are huge number of tools. So these days, Python has some library. Python has some packages. R has some libraries which does really good metabolomic interpretations and analysis. Right. So you can use Python. You can use R. You can use MATLAB, so etc. So they have very well defined packages and libraries that are available. So Metabo Analyst is one very useful tool for metabolomic analysis, right? Metabo Analyst, right? So it has a huge number of applications, sorry, features, which you can use. This is one research that has been done by one of my students. So what she has done taken is like she has taken the breast cancer data. So breast cancer is one of the very most diagnosed disorders or cancers observed in females. So what this project is was all about is to find how metabolomic analysis, particularly metabolic pathway analysis, using metaboanalysis software can help in the diagnosis of breast cancer. So the data set that is being used for the analysis uh, was taken from metabolomics workbench. Metabolomics workbench that I mentioned is the data set available various data sets of various diseases. So she has taken this data set of 134 samples and 272 compounds from the metabolomics workbench with different concentrations of the metabolites. The dataset was acquired from City Hope Hospital in US and was further pre-processed using metaboanalysis. Compounds that has been seen that has increased concentration by analyzing by doing the analysis was homocystic acid, methyl pyridine, which are quite in link with the with the has been potential biomarkers of breast cancer. Fine. So metabolic pathways, such as even pathways that has been observed here are amino acyl tRNA biosynthetic pathways, alanine, aspartate, glutamate. So these are some pathways which are actually distorted during cancer in cancer patients, but it has been like she has actually identified using metaboanalysis by using the data set from metabolomics workbench. So these box plots have been and kernel density plots have been box plots and kernel density plots have been generated before and after normalization. The box plots show at least 50 features due to space limits, and density plots are based on all samples. Selected methods are that have been used are row-wise normalization, normalization through sample median, data transformation, cubic root transformation, data scaling, and auto scaling. Univariate analysis she has performed, not multivariate analysis. Total of 193 significant compounds has been identified and 79 insignificant compounds were determined by one-way ANOVA method in metaboanalysis itself. So everything is integrated in metaboanalysis. It's a very useful and wonderful software for performing data analysis of high throughput data analysis of metabolomics data. Fine. So multivariate data analysis has also been done later on because as not much of the significance of the data was returned because in the previous univariate analysis. So multivariate analysis was performed, principal component analysis was done. So you can see these score plots um, in principal component analysis. This results in a pairwise score plot and its respective 2D and 3D model that represents the overall the overview of the various separation patterns among the most significant PCs, where PCs are principal components are the eigenvectors representing the direction of this. So you know all these things. So this the 2D model is a score plot. So 2D model you can see is a score plot. Uh, uh, with PC1 and PC2, representing two clusters, one cluster with control group and another with the three. So this is all clustering that has been done in metaboanalyst itself. So you can do that, all these things in metaboanalyst itself, right? So it has been done, this clustering has been done on the basis of the three stages of cancer. Fine. So the results from pathway analysis. This is another important thing that we do. We can do is the pathway analysis. So the results from the pathway analysis are presented graphically. You can see here the third one. The structure of the biological pathways represents our knowledge about the complex relationships among molecules within cell or a living organism. The figures that says you can see here represents the overall impact on the pathways on moving the cursor. So if you move the cursor on these various yellow and orange and red dots, red bubbles, you can actually see the impact on the pathways. Fine. For example, metabolites which are affecting the amino acyl tRNA biosynthetic consists of L-aspargine or phenylalanine, etc. You can see here by moving the cursor on it. Metabolomics analysis has been identified as one of the most useful method for the diagnosis and in-depth study of any particular disorder in humans. I think I have mentioned this quite a many number of times so far. So I guess let's move ahead. Metabolomics data analysis using machine learning approaches. This is done by another student of mine who has worked on vitiligo. So vitiligo data was not very easily available. So we have asked this research group in Iran. They have provided us with the data. So what is the difference is that this data set is basically the previous one, breast cancer, was a peak data set, that is the spectrum, right? And this data set is MZ values, right? So in vitiligo. So these are the two types of experiments that we have done. So here we have used using we have used R basically to do this analysis, and I think ma'am has talked about this generation of heat maps in R data collection and statistical analysis. The data which have been utilized in this study was collected from the individuals who has come and consulted in the hospital in Iran, and it was done in 2018. The datasets provided has been standardized in such a way that it only the vitiligo patients with more than 25% of skin area affected with depigmentation has been were classified as vitiligo and rest were classified as control. Right, so the data contains an appreciable age range and the gender distribution is almost equal. The distribution of healthy and vitiligo is almost equal across the age ranges and gender. This also elucidates on that fact that vitiligo pathogenesis is independent of age and gender. So this is one of the uh, uh, inferences that the dataset contains the details like the patient's ID, which have been dropped, which we have dropped, which have not taken the patient IDs. It further contains the age, health. So these are the different aspects of the data that we got in the Excel sheet: age, health, and levels of 22 amino acids in the blood of all the 22 amino acids. So after data acquisition and processing, we have conducted statistical analysis on the datasets to have a better understanding of the various features. So this is like logistic regression has been run. KNN uh, has been run. These are the values that we got: random status values, accuracy. We got good accuracy, but we need more work. SVM algorithm has been used, KNN and SVM algorithm, but we need more work on it. So anybody interested who wants to work in this area are most welcome. So I wanted to see more patterns in it right now in the data, if possible. If anybody is interested to do machine learning in metabolomic data in this vitiligo, uh, anybody is most welcome, right? So these are some of the book chapters that we recently published on metabolomics, flux balances, metabolomics, etc. And thank you. So I think I have like run through the whole very quickly through the presentation.
Thank you so much, sir, for such a wonderful session. It really provided an in-depth knowledge. So Gauri Panajkar says, sir, please share the form link again. Okay. I would encourage the participants to share any doubts that they have over the chat box.
But no questions. Any question, if anybody has, please you can ask. I'm not able to.
I don't think there are any.
Okay. So if you have any questions, I'm here in Amity University, fourth floor, J3 block, and our lab is here, the systems biology and data analytics lab. So anybody interested to work on multi-omics analysis, multi-omics data analytics using various methods, you are most welcome. Thank you.
All right. I don't see any questions in the chat box. So I'll just make a small announcement for everybody and Sharmila to answer your question. So for today, we do not have a quiz. I've shared a link where you are supposed to be submitting the results of the assignment that you do. So you can either write the steps in the form of a Word document and upload, or if you want to paste it there in the link itself, that is also okay. It is not that your certificate is based on this assignment, but we encourage everybody to submit the assignment. There are no marks associated with it, but you please try and submit the assignment. There are just 3 questions. One is on ensemble, which you did on the first 2 days, and then second question is a metabolomics, a practical kind of a question where you can go ahead and try any database, and the third question is microarray data analysis with GSC 21222, which I just showed in my class. So what you can do is just create a Word file maybe, and just write the steps and the output that you're getting and paste it there. Just a screenshot will also work. So you can do that and submit it. So we are going to be closing the session in the next 2 minutes or 3 minutes. Please take down the links to the quiz. And in case of any query, you can please write to me, or you can write to CCBB email that you have received for your registration. So if you need my email for any concerns, it is pnarat@amity.edu, which is also written in the chat box. I have reshared all the links today. So please make sure that you attempt everything. I think then we are all set to end the workshop. If there are no questions, then we are good to go. This link will be open over the over today.
I have one thing to say. So out of the participants, the external participants or internal participants, I want 1 or 2 participants to say their feedback remarks. Please, you can switch on your cam. I don't think so. You can switch on your camera. You can just say a few words about the workshop. That would be helpful for us. Anyone who wants to voluntarily come and say a few words? Acha, okay, you can only write on the chat box. I guess you are not. Yeah.
Anybody wants to write, then I can, as a co-host, like unmute them so they can write over the chat box that they want to speak.
Okay, so I'm so sorry. I was not aware that they cannot say anything. So no problem. I hope everyone enjoyed the session thoroughly, and we will definitely come up with more such interesting workshops in coming months or coming in the next year. So thank you so much for your attention. Thank you.
There is some feedback. It has really been a wonderful session. Thank you so much.
Thank you, Sati, and thank you, Barkha, and thank you, everybody. So please take down the links. Thank you so much for the encouragement, and we do hope we can continue to deliver more workshops in the future also. So thank you, IT. If you're there, I think we are good to close.